In this investigation, we want to prepare a wet mount to show the structure of the epidermal cells of onion. So this very thin membrane is what I'm going to remove now by pulling it out carefully from where it is attached to the rest of the onion, and I am going to put this on a pre-measured piece of paper. I want to get a square of approximately 1 centimeter in diameter. I've already cut some squares here. I'm making 2 mounts, but what I'm going to demonstrate to you is the first one that I'm making using iodine solution, and rest this over my template square so that I can get the approximate size. Use this to get the piece that I want. So now I have a square of my onion epidermal layer, and I'm going to rest this square onto a glass slide and just flatten it out a little bit. So now my square is resting on the glass slide, and to that I'm going to add a drop of iodine solution. I'm adding this drop of iodine to my onion so that I can tell whether or not there are starch grains within the cells of this piece of onion epidermis. It's a plant, and therefore it would have carried out the process of photosynthesis, and one of the main products is starch. Iodine is a solution that we use in the lab to test for the presence of starch. So I've added 1 drop of iodine. And I'm going to tilt the slide slightly and allow the drop of iodine to pass over all of the parts of the onion epidermis that I have put on my slide. Once I'm fairly sure that the iodine has actually contacted most of the slide, I am going to use another little piece of tissue and dry off the excess solution. Put that aside. Next, I'm going to take a cover slip, which is a very thin glass, and I want to lower this cover slip over my piece of onion. So what I do is I rest the cover slip on the slide like that, and I'm using this pin that's in my right hand to gently lower the cover slip down onto the onion. I use the pin again to gently squeeze out any air bubbles that might have become trapped under the cover slip. And now I have a wet mount of onion skin on the slide with the cover slip over it. So my next step is to do a similar preparation but using water instead. But what we're going to do next today is to take you to the view of my onion. So I have put this slide onto the microscope stage, and I'm going to use the lowest power times 4 power to find the image first. And I can see that there are lots of epidermal cells in this image. You can see all the cell walls very clearly, and you're seeing all the connections between the cells quite clearly. And now I am going to change the magnification to a slightly higher power, times 10, and take a look at our cells again to see whether we can see any greater detail within the cells. And we can see inside of the cells, you're seeing the cell walls very clearly outlined. They appear to be much darker than the cytoplasm inside of the cells. With this magnification, however, we can just barely see some of the starch grains that have been stained by the iodine solution, but we can't really discern clearly any of the other organelles within the cell. You would probably need a microscope with much higher resolution in order to see things like the nucleus and the chloroplasts a little bit more clearly. But right now, what we can see are definitely the cell walls. We are seeing the connections between all of the various cells. It forms a sort of brick-like pattern. And we can see the material within each one of the cells. We are seeing the cell cytoplasm, which is, it has a slightly grainy appearance. So the little dots we are able to discern in there could represent any of the typical organelles that we would find within plant cells, such as the chloroplasts, the nucleus, mitochondria, etc. But we can't really tell which ones we are looking at using this particular power. Under the highest power for this microscope, it does not become much clearer. So we are going to end our observation of these onion cells with a view where you can see what the cells look like using this light microscope, but just at times 10 of the objective power.